Hello everyone and welcome to this Q&A session with our speakers. It's my pleasure to be here today to moderate this Q&A, and we're going to ask all of your questions to our speakers here today. We have Claire Jones with us, Chief Scientific Officer at Talisman Therapeutics. We have Stefan Przeworski, Professor at Durham University. We also have Darius Wadera, Associate Professor in Stem Cell Biology and Regenerative Medicine at the University of Reading, and Heiko Werdak, Head of Stem Cell and Brain Tumor Group at the University of Leeds. So we'll get started, and my first question will go to Claire. So Claire, how do you achieve a fairly equal distribution of microglia in your co-culture models?
So it kind of happens spontaneously, actually. When we generate the co-cultures, we make the neurons and the microglia separately. We culture the neurons and maintain them in monoculture up to about day 50. And then we add microglia and they migrate into the microglial cultures. So actually, we don't really do anything specific other than maintaining the cells and obviously making sure that we're treating each well the same. But actually the microglia kind of infiltrate into the neuronal culture and distribute themselves in that kind of really nicely tiled fashion on their own.
Fantastic. That's great. Thank you. And my next question, it's a question to Stefan. So Stefan, what are the main challenges facing companies that are interested in adopting microphysiological tissue models?
Well, there's several things one needs to consider. Firstly, the first thing I say is there's no single solution. There are lots of different technologies out there on the market. And the first thing is the question that the end user is trying to address. So they have to define that. They then have to look to see if the technology is actually available, because they might have read about it in a paper, but doesn't mean it's commercially available. So they need to establish that. Then they also need to consider the throughput, so the number of samples that they want to look at, the endpoint analysis. So are the techniques and assays that they have got in mind, are they compatible with the technique? The methodology, the cost, all those sorts of elements need to be considered. But they also have to bear in mind that it is more work, it is more expensive, it may take longer, but the benefits are that you potentially might get more informed, reliable data, which is more in vivo-like, as opposed to a model which is conventionally in 2D, which, as we know, has significant limitations. So there's lots of elements to consider.
Absolutely. Thank you very much for that. And my next question will go to Darius. So are you planning on applying hydrogel scaffolds to different applications in the future?
Yes, indeed. At the moment, we are pretty much focused on optimizing the composition of the hydrogels in order to modulate the potential of the secretome of the cells. So as you might know, mode of action, specifically of mesenchymal stem cells, it's not that much that they integrate and differentiate. They modulate endogenous regeneration in the animal or in the model. So what we are trying to achieve is to increase the potential of the secretome by applying very specific 3D cell culture technologies in combination with biophysical stimulation, more specifically with certain parameters of electrical stimulation. And this can be pretty much applied to different end applications. Currently, we are looking how the secretomes actually influence cancer cell behavior in terms of modulation of apoptosis, proliferation, and migration. Again, this is fairly broad, and that's what we're doing at the moment.
Fantastic. Thank you so much for that, Darius. And Heiko, could you tell us what are the main differences in your work between assembloids and tumor organoids?
So the main difference is what we would call an organoid, and it's quite a fussy nomenclature between spheroid and organoid. But what we think is an organoid is if we take patient-derived cells, fresh from surgery, we do a mechanical, slightly enzymatic dissociation, and we put this into a hydrogel, matrigel, or any type of scaffold. And then this matures. We can put this in a spinner flask, and this is a one compartment disassembly of a tumor and then reassembly of a tumor. We have a microfluidic chip environment that we are testing, for example, for these applications. Whereas the assembloids, this is based on this observation that the cancer cells have the capacity to invade neural environments such as cerebral organoids. And then you have a two-compartment system, which we find very attractive, for example, in selectivity assays, where you see this real-time behavior of invasion, and you can perturb this. And you can then, at your endpoint, it doesn't have to be image-based, so we are also taking our cells, the cancer cells, out of the environment and do single-cell analysis at gene expression level, for example, where we can see the environment cells and the cancer cells. I hope this explains the difference.
That's fantastic. Thank you so much. And that brings us back to Claire. So Claire, could you tell us, do you experience heterogeneity in your IPS-based microglia cultures?
That's a great question. So we've looked at this quite a lot, as you can imagine. They're reasonably homogeneous. So we're kind of aiming for sort of 80% plus homogeneous cell populations. We have a kind of quite a rigorous QC procedure. So we check for expression of various different markers, and we always check that we're getting decent responses to LPS as a kind of baseline. And if we don't see that, then those cells go in the bin, and we just assume it's a bad induction. So it's pretty good heterogeneity at the population level. If the question is asking whether or not on a cell-by-cell basis there's heterogeneity, that's a kind of work in progress. And my prediction would be that, yes, we will see heterogeneity, but that is likely linked to the environment that they're in. So I think we're quite confident that our protocols are good at generating a homogeneous starting point. And it's actually the microglial progenitors that we add into the neuronal culture. Once they're in with the neurons, if you take a snapshot time point, you will see heterogeneity, because even morphologically, you can see that they're just responding to what's going on around them. So that kind of half answers the question.
That's great, thank you. And that brings us back to Stefan, and I have a statement here and then a question. So the attendee said, it's really interesting you see a difference between young and aged cells, which you incorporate into the skin models. More generally, have you seen any differences between human cell donors in these models? Is it reproducible and robust?
So the aged skin models use cells from donors of different ages, young and old, and then we can create young and old models. They are robust. The differences we see are when we first of all expand cells in 2D culture to generate the cell numbers, and we then look at the proliferation rates, and we see differences in the proliferation of old cells versus young cells, old cells being slower cycling. But between the groups of old and the groups of young, there are quite a lot of similarities and consistencies. We then produce our models. We then see the phenotype, as I explained during the talk, so thinner epidermis and less ECM. So all of that is possible. The reason we believe for having these differences is partly to do to the numbers of senescent cells and the bystander effect, which is a signaling effect via SASP, as it's known, influencing the epidermis. So we can take old dermal cells and put them in underneath a young epidermis, and we see the young epidermis thinning. So that demonstrates the signaling between the two compartments. So it's very versatile technology to enable you to research this process. And you see, characteristic phenotypic differences of the aging process during that.
Fantastic, thank you very much. Darius, a question for you. What imaging assays did you use to assess the PEDOT, PSS, and collagen constructs?
That's a very good question, and indeed, imaging in PEDOT-PSS is quite tricky since light microscopy can't achieve that much. So you rely on the fluorescent signal. So all the imaging needs to be indirect. So what we used to do is either label the cells transplanted, or cultivated in PEDOT-PSS with PKH67, which is a membrane dye. You can also transduct them with the lentivirus, for example, expressing GFP of RP. That's the only way to go forward. And indeed, if you assess, for example, differentiation of those stem cells, you can't apply traditional methods relying on light like alizarin red to assess mineralization. It's not possible in those scaffolds. So this is certainly a drawback.
Okay, thank you very much for that. And Heiko, could you tell us, how do you incorporate the characteristics of your patients into your disease models?
So they are inherently in the patient material that you dissociate and then use to create assembloids. So these are, the system works with models where we take the tumor cells and we make, first of all, 2D cell cultures that have similarity with neural stem cells. This is in the brain tumor field. These type of models have been used since 15 years, and there are resources, and they are pretty good models. But it also works with tissue pieces, which is even more exciting for us to not go through a 2D step, but directly assemble round tumor pieces with the mini brains or neural environments. So this is how the patient characteristics get into this, what we use as a scaffold of a 3D model environment.
Thank you very much for that. That's great. And I have a final question here, which will go to Darius, and then unfortunately we'll have to conclude our Q&A. And that is, how do you explain that the doubling time decreases with fibrin-based scaffolds? Sorry, Darius, you're on mute.
Apologies for that. This is a very good question, and this reduction of population doubling time is not exclusive for fibrin matrix. This is what we observed with this type of cells, regardless of the scaffold we have used. We tested population doubling time and basically proliferation in fibrin, collagen. We also tested peptide-based hydrogels. They basically speed up regardless of the scaffold if you put into 3D. Instead of 2D. So this is quite general phenomenon and not exclusive to fibrin.
Of course. Thank you for explaining that. That's great. And thank you very much to all of you and all of our speakers for both your presentations earlier today and also for joining us again for this Q&A. If anyone had any questions that they weren't able to get to us in time, then I'm sure that our speakers would be happy to answer these, via the platform or, via email. They'd be happy to connect with you. So thank you very much again, all of you, for joining us.